Good morning, and welcome to the second session of Health Matters. My name is Usha Nesimoni. It's my pleasure this morning to introduce Dr. Anne Brunet. Dr. Brunet is a professor of genetics here at Stanford University. She obtained her Bachelor of Science from École Normale Supérieure, I hope I'm saying that right, in Paris. Her PhD is from University of Nice, and her postdoctoral research training was actually at Harvard Medical School. Her research focuses on age and longevity, specifically as it relates to the nervous system. She's published many peer reviews, papers, and book chapters, and she has received numerous awards, too many to actually cite today. Please join me in welcoming Dr. Anne Brunet.
All right, so good morning, everyone. And first of all, thank you very much for the organizers for inviting me here. It's an honor to be here this morning, and thank you very much for this very nice introduction. So today, I'm going to talk about aging and longevity. So as nicely captured by this drawing from Leonardo da Vinci, there are obvious differences between a young individual and an old individual. But as we all know, aging is also accompanied by the decline in many physiological functions. So in fact, this has been graphed by scientists, and you can see here that aging is accompanied by decline in fertility and pretty much in the decline of every tissues, whether it's the muscle, the lung, the kidney, the heart, and even the brain, which by this global measure might seem pretty intact. In fact, as we all experience, if we look at high cognitive function, such as memory, working memory, long-term memory, speed of processing, and etc., this really strikingly declines with aging. And it starts actually pretty early on in the 20s and 30s, and it keeps declining. Now, you can also see that verbal knowledge stays approximately constant and even increases, but this is, scientists think it's due to experience and exposure to more words. So really, aging is accompanied by a decline in many functions, including high cognitive functions. Now, aging is not only accompanied by a decline in normal function, it's also associated with a constellation of diseases. In fact, you can see on this graph that heart disease, type 2 diabetes, pneumonia, and you can see this also very exponential increase in neurodegenerative diseases such as Alzheimer's and Parkinson's. So this really raises the fascinating question of whether there could be, in fact, a common cause behind all these diseases, aging. And in fact, aging is actually the primary risk factor, the main risk factor for all those diseases. So maybe a new view is that instead of, in addition to treating all these diseases by their specific symptoms, if one were to understand the root cause of aging, one could have a leverage to perhaps change those this constellation of age-related diseases. So really, the fundamental question in the aging field is the following. Is aging just a downway slope towards decline, disease, and death? Or is there some hope, or is there some plasticity to this system? And can aging, in fact, be manipulated? Before the past 20 years, aging was thought to just be a byproduct of wear and tear, and not to be something that one could act on potentially. But this view has completely changed. And in the past 20 years, it has become clear by the work of many, many, many scientists that aging is not just an inevitable, inexorable byproduct of wear and tear, because there are specific genes that can alter the course of aging. there are also environmental factors or even specific compounds that can, at least in model organisms, alter the course of aging. And then finally, there are some studies indicating that some blood factors can even revert some hallmarks of an already old organism. So at least in the lab, aging can certainly be slowed and even reverted. And in the next 3 slides, I'm going to give you some examples of some of those genes and environmental factors and blood factors. As for genes, it has become clear, for example, by studies of people with exceptional longevity called centenarians, people who live more than 100 years old, or super centenarians, people who live more than 110 years old. So those people are not very numerous, but they exist. And here you have Jeanne Calment. She actually turned out to be French, and she's so far the longest person who was recorded in all humanity as being the longest lived person of the world. She's dead now, but she got to live up to 122 years. And studies now in centenarians and supercentenarians have revealed that there exist some genes that those centenarians or supercentenarians have that can protect them from many of those, the constellation of those diseases. And I'll come back to those genes in the middle part of my talk. So this is for genes, but let's say we don't all have those good genes in our families. So what about environmental factors, things that we can more easily act on? Well, it turns out that in the past 10 years or so, there has been experiments, again, in many model organisms that have unraveled environmental factors from the outside that can have a profound influence on longevity. And in fact, it is thought that the proportion of longevity in humans that is determined by environmental factors is 70%, whereas 30% is determined by genetic factors. So the environmental factors that in the lab, and presumably some of them in humans, promote longevity are the following: exercise, dietary restriction, so actually a pretty strong restriction in dietary intake. It's pretty hard to do because it's 1,200 calories per day, the equivalent in humans. Rapamycin, those are compounds found in the soil of Easter Island. So rapamycin is one of those compounds. Metformin, it's a compound that comes from French lilac. And resveratrol, probably several of you have heard of it. There's a compound that's present in the skin of grapes, for example, and that's present in red wine. So those compounds, at least in model organisms, have the property to extend, and for some of them, even in mice, rapamycin has been shown to pretty significantly extend the lifespan and the youthfulness of mice. And there are currently trials in humans for rapamycin. Okay, so those are for compounds. What about this amazing potentiality to reverse an already old organism, or hallmarks or characteristics of an already old organism? Well, studies here by colleagues at Stanford, for example, Tom Rando and Tony Wiscory, have shown that you can, in the lab, experimentally fuse 2 animals, one young and one old. You can make Siamese if you wish. The mice are fine, actually, by doing that, but you suture them by the flank, and they wind up sharing a blood circulation in about 3 weeks. And in fact, what this has shown is that characteristics of aging in the old mice, they have an old heart or old muscle cells or an old brain, get rejuvenated when they get hooked up by the blood circulation to a young mice. And this has been followed by the fact that the simple injection of blood, repeated injection of blood from young mice into old mice also recapitulates some of those rejuvenating factors. So this indicates that there are circulating factors in the blood that can have, at least to some degree, some rejuvenating potential on an already old organism. And it's not just a matter of slowing, but there can also be some reversal of some of the aging features. Okay, so those are for the elements, whether genetic or environmental, that controls the longevity. But a key question also, when one thinks about that, is that which cells in the body are important or responsible for longevity or are susceptible to aging. And we, as well as many others, have postulated that there might be some cell types in the body that could be particularly interesting for aging research. And some of those cells are what's called the stem cells in the tissues. So I'll explain what stem cells in tissues are, but suffice it to say here that stem cells are cells with amazing regenerative potential. They can basically renew themselves almost ad infinitum. So in our lab, and it's also done in other labs, we are interested by two fundamental questions as it pertains to those regenerative cells. The first one is, could preserving or tapping into the stem cells that we all have in our body be sufficient to extend the health of the tissue, the regeneration of the tissue, and even the lifespan of an organism? And second, because their stem cells have this regenerative potential that other cells do not have, can we almost use them or understand how they do that to understand better get new insight into the mechanisms of rejuvenation or regeneration, because they may have something, those stem cells may have something unique that they can do that other cells cannot do, and this could be very informative. Okay, so what are those stem cells? As I'm sure you guys have heard of that, we actually, in pretty much all our tissues, even as adults, still have populations of cells that have amazing properties of what's called self-renewing, so regenerating themselves, that's why the word stem, but also to give rise to the differentiated cells that are very characteristics of our body, muscle cells, heart cells, brain cells, and etc. So they have this dual property of regenerating on the one hand, and also being able to reconstitute cells in the tissue. So those are two very important characteristics. If we take the brain as an example, it was thought for a long time that the brain had no regenerative capacity, because the common knowledge is that we have the neurons that we are born with. Well, that's not completely true. It is true that most of our neurons are the ones we were born with, but the adult brain, this is true in mice, but this is also true in humans, contains pockets or reservoir, if you wish, of stem cells in an adult organism. So we all have this reservoir of stem cells in our brain. And those stem cells, called neural stem cells in our brain, they are in two regions of the brain, and they can self-renew, so renew themselves as stem cells, and they can also amazingly give rise to all the three key cell types in the brain. Neurons, of course, very important for brain function and memory. Astrocytes, that's the glia, that's also very important supporting function. And oligodendrocytes, which are also very key because they form the myelin sheath around the neurons, and they allow proper connection between neurons together. It has been shown that those neural stem cells, those pockets of cells, even though they are not very numerous, are very important for the function of learning and memory, because if they are removed genetically, if one deletes them specifically, the animal is less able to learn and to remember. Okay. Now, the key question then is that those stem cells are a really nice reservoir and a nice resource to tap into, but what happens to them during aging? And unfortunately, it's not good news, but that's what people are trying to understand. During aging, suffice it to say that there is a drastic decline in the ability of those cells to self-renew. That's on the right side of the graph, as well as in the ability of those cells to give rise to new neurons. And that's on the left portion of the graph. So really, during aging, there is a decline in adult stem cells that seems to precede the decline in tissue regeneration and the ability to repair the tissue. And for example, in terms of the brain, those stem cells are very important for after injuries such as stroke to perform some of the repair. And that their decline during aging may explain why it's so much harder to recover from a stroke when one is older than when one is younger. Now, if this wasn't enough, that there is this drastic decline in stem cell function and tissue regeneration during aging, there is another problem with stem cells during aging, which is that they become more, they have this regeneration potential, and that's great, but that also makes them more at risk for specific mutations, and in particular mutations that then could give rise to cancer. And during aging, stem cells become more prone towards having those mutations. So this is the double whammy of aging, where the normal function declines and the propensity to give rise to cancer, to those cells to give rise to cancers, increase. So really, it's fundamental to understand how to preserve and protect these cells. So the questions we've been having in our lab is how to preserve those cells and how to maybe even tap into this potential during aging and even reactivate the pool of old stem cells. And one idea that we had was perhaps genes, the genes I was mentioning before, that can promote longevity, the genes that have been identified by their capacity to extend lifespan in humans or other organisms, could have an important role in stem cells. And we could use that as a handle to reactivate some of those cells. Okay, so this gives me the opportunity then to discuss the genes that are important for longevity, because there are several of them. And in fact, despite the fact that longevity is regulated only 30% by genes, it's actually very interesting to understand this genetic underlying, because there is probably a lot of things that can be done at the interface between genetic and environmental factors to understand this. One key discovery in the aging field dates back to 20 years ago, where in fact the group of Cynthia Kenyon at UCSF, she's now at this company funded by Google called Calico, identified a mutant in the little worm. It's a little worm called Caenorhabditis elegans that lives in the soil. And in this little worm, what Cynthia Kenyon found was a mutant that could live 2 to 3 times longer. And that's what you see on the graph, that the mutant worm in black dots can live 2 to 3 times longer than a normal worm. And not only could it live 2 to 3 times longer, but it was useful for 2 to 3 times longer. It wasn't that its decline period was extended. It was really its usefulness period that was extended. Now you could argue, well, sorry, I mean, it's just a little worm that works lives in the soil. How does that relate to us humans? But in fact, in the past 20 years that this discovery was made, it has been amazing because it has gone from worms, and it's been shown to be true in flies, in mice, and even humans. So there's really conserved, even though animals are very different in their lifespan, and a little worm lives 20 days, whereas a human can live up to 120 years, some of the circuitry, genetic circuitry, is fundamentally conserved. So that's really interesting. For molecular aficionados, the way it works is very actually beautiful in the sense that if we have circulating insulin, if we have insulin, let's say after a meal, and it connects nicely to environment. When we eat a lot, there's a lot of insulin that's circulating in the body, and that's both good and bad. It's good because it helps, obviously, with dealing with the food, but it will activate a pathway within the cells that winds up inhibiting a family of longevity factors called FOXO, and that's what you see in red. And those are conditions that are actually associated with aging. Now, if you do dietary restriction, or that was the mutant in those little worms that live 2 to 3 times longer, if you have a mutation in that pathway, this factors called FOXO is no longer sequestered like it would be by insulin. And it can go, it's a pro-longevity factor, so it can go into the nucleus of every cells. And in every cells, it can trigger what's called the program of genes that are important for many functions of detoxification, repair of damaged DNA, proper folding of protein. So it really has a positive function on the cells. And what's truly remarkable is that this longevity pathway that connects, it's a genetic pathway, but you can see how it connects nicely with the environment because it integrates signals from the blood, like insulin, that are themselves responsive to food and exercise and etc. This pathway has been recently shown to be actually one of the genes that's critically different in the centenarians or supercentenarians. So that's an example of the paper here. But if you guys have done 23andMe, for example, it's on, you can look at your chip panel in there. I don't have any stocks in 23andMe, so it's, I'm just saying. So if you have in the FOXO3, in your FOXO3 genes, at a specific position, if you have TT, those are like bases in DNA, that is associated with normal lifespan. But if you have a TG, other base in DNA, or even GG, then it's associated with an increased chance of becoming a centenarian and also higher cognitive function for longer. So that's quite interesting. So the questions that we were wondering then in the lab is that if we go back, that was about genes, but if we go back to the question about cells and the stem cells, we asked the question then, well, what can this amazing pro-longevity transcription factor FOXO that is present in centenarians under this variated form, what does it do on the stem cells in the brain? Of course, we use mice for doing those experiments. And what we have shown is that if you make mice, if you generate mice that have a mutation of this pro-longevity transcription factor in their brain, the stem cells, their stem cells, become as if they're prematurely old, if you wish. They exhaust prematurely, they can't self-renew very well, and they become skewed, in fact, towards giving rise to astrocytes, a lot of astrocytes, at the expense of giving rise to important neurons and oligodendrocytes. So really, when cells, neural stem cells, are missing this pro-longevity gene, they behave as if they are prematurely old. So what that means is that those are stem cells, and you can see the neurons, beautiful. So the FOXO transcription factors can really preserve and even push those stem cells to form new neurons. And the new neurons, you can see them, they are in green here in this picture. At the molecular level now, we have a handle, because this tells us that this FOXO is really fundamental for preserving stem cells. So we've been curious, and again, it's a lot of molecular and genomics and genetics, but that's what Stanford is really super great about, is that it allows you to have an unbiased view then in the genome and of the cells. And what we were able to find are all the genes, by using these amazing technologies that Stanford constantly develop, all the genes that this pro-longevity transcription factor can regulate in the cells. And it's about 2,000 genes. A lot of new genes, so that means a lot of potential to understand better how to activate, to reactivate, and how to preserve the pool, the reservoir of adult neurostem cells. And in the Q&A session, I'll be happy to discuss some of those genes if you're interested. Okay, so that was for the first aspect that I wanted to discuss. And in the last part of my talks, I will present to you things that we do in the lab, but that are more forward-looking, or trying to understand in the future what are the future of aging research. So one aspect that we've been particularly excited about is this idea that perhaps there is some quote-unquote secret to immortality in the stem cells, in their ability, like the stem cells themselves have figured out, if you wish, a way of being immortal themselves, of having this amazing capacity to self-renew. So can we, at the molecular level, dissect this function and perhaps understand better the molecular characteristics of usefulness? And one particular aspect that's fascinating in many, many ways, but I will tell you the way it is fascinating for aging, is a process called reprogramming. I'm sure you've heard of it. It's very highly used now. Reprogramming is the process that solely by adding 4 transcription factors to the cell, to differentiated cells, an adult cells, can convert it back to an embryonic-like stem cells, which is called iPSC, which stands for induced pluripotent stem cells. So this process has fascinated many people in the world, but probably more, way more, for its de-differentiation aspects, because it turns a differentiated cells, a muscle cells, a brain cells, back into a non-distinct, like a very embryonic-like cells with a lot of embryonic potential. So a lot of people have been fascinated by this. We are fascinated by the fact that if you think about it, this process almost mimics, it's the ultimate rejuvenation process, because it's been designed, in fact, to mimic the fertilization process. And 2 actually pretty old parents can have a baby, and the baby is born young. It's not born old. So there is a process, an amazing process. I mean, it sounds obvious, but it's not that obvious, because the gamete of an older organisms have been subjected to a lot of accumulated damage around the time. Yet the babies that are born young. So there is an amazing process in the process of passing through generation that has this reset property. And this process in the cells mimics it. So we are very interested in the future to understand how, molecularly speaking, this reset occurs, because perhaps it's possible that it can give us some new insight into what is important to remove some of the accumulated damage, some of the bad aggregated proteins, or etc. in the cells of an adult organism, not just its gametes. All right, so that's the promise of iPS technology that people use for regenerative medicine. It has many promises, including, as I said, the process of rejuvenation, but obviously also in the clinic to do cellular replacement therapies for a lot of age-related diseases. So many promises for this technology. Okay, and I'd like to end my talk by something that we're developing here new at Stanford, which is to develop new model in which we could rapidly, very rapidly, screen for new genes or new compounds that affect aging. And in fact, if a human lives very long, so it's very hard, obviously, to have the results, or even a mouse lives very long. It lives like 3.5 years. So in order to screen rapidly, it's actually pretty hard. So what our lab has decided to do is to generate a new model for aging research that was compatible with screening, was very fast on the one hand, but also had the characteristics of a mammalian system and had organs and tissues, because the worm is nice, and people use it, but it's very wormy. So, so we've decided to develop here a fish. So granted, it's not a furry, it's not a mammal, but it's a vertebrate. So like vertebrate, it has a heart, it has a brain, it has stem cells, which worms don't have. It has pretty much it has an immune system. It has pretty much all the characteristics, because the branching out, evolutionary speaking, is pretty recent between vertebrates, between fish and mammals. So it has all the characteristics of us. And what's amazing about this fish is that it's a naturally short-lived organism that we went to Africa to capture. So it lives in those ponds of water in Africa. And those is in Zimbabwe and Mozambique. And those are exactly the same ponds in the wet season and the dry season. And in fact, this fish lives an amazingly naturally compressed, it recapitulates our lifespan, but in super fast period of time. And it has to, because the ponds completely dry up. So in 4 months, it basically recapitulates a very short lifespan. Now we bring it back in the lab. So in the lab, we work on fish, on mice, on all sorts of different organisms, because we feel like it's really by having different models that you can understand the complexity of aging. So those are the fish. And what's nice is that this fish, even in the lab, it lives, well, just across here, it lives 4 to 6 months. So it really recapitulates this very short lifespan. And now we can do experiments and screens and screens and screens for things. And even in this short lifespan, sorry, it does age. So it has signs like we do. It has signs of fertility decline, muscle loss, learning decline, the neurodegenerative deficits, cancer, and etc. So it's a really good model. And what we've developed recently, and we published that at the beginning of this year, is a complete genomic to phenotyping platform here. And this is what's, again, very awesome at being at Stanford, is that there's all these new technologies, and we sequence the genome in very rapid manner. We were able to sequence the genome and edit, meaning modify this genome, so that now we can pretty much target all the genes, mimic human mutations, human variants of centenarians or human variants for a specific disease in this fish. And then we have a model, basically, for our disease or for aging. And we can now screen for drugs that reverse it, and etc. So basically, this is the life of a human. Of course, it's very long, 120 or 100 years. And the lifespan of the fish, and it's much shorter. It's compressed. And this really gives us this opportunity, we think, to screens and also to discover new biology about aging and diseases. And the goal is to really understand how aging occurs, how health can be preserved, and also how to delay a lot of age-related diseases so that we can, as humans, live the healthiest life, the longest time as possible. So with this, I would like to acknowledge the members of my lab here at Stanford, Stanford University itself, for providing amazing ability to do research and discoveries. And with that, I would be happy to take questions. Thank you. Questions?
Thank you. At the beginning of your talk, excuse me, you mentioned 3 compounds that were related to aging, and then you discussed the insulin FOXO pathway. Are there any connections between those compounds and that pathway?
Yes, so that's a great question. And in fact, there is. So this insulin FOXO pathway, one branch it has is called the mTOR pathway. So insulin can also inhibit FOXO on the one hand and activate TOR, the TOR pathway on the other hand. TOR is mTOR. It's a protein kinase. That is that's the proximal target of rapamycin. So in fact, when you block rapamycin, you actually block the insulin pathway and wind up activating the FOXO. So yes, it is connected. It's basically taking rapamycin would be the equivalent of doing dietary restriction without doing it.
I have a question. You mentioned in your talk that this lady who was 122-year-old, she had those particular genes. So my question is the current technology where you can screen people if anybody wants to see if they have those particular genes for longevity? And if so, what is the cost of such screening?
So again, disclaimer, I do not have any interested things in 23andMe, but 23andMe does have one of the genes. It's not, all the genes are not known, and there is also probably gene interaction. There are some alleles that are associated with longevity for sure, but it's also possible that if those alleles are present in an individual that has risk allele for a very strong disease, they may or may not be sufficient to counterbalance them. So I think we need to, before like the screening that you're talking about is feasible, one needs to understand a little bit, a lot more about the genetic interactions with the gene. But in terms of the costs, you know, sequencing a genome those days is actually becoming cheap, a full genome, in fact, is becoming cheaper and cheaper, and probably with $1,000 or $5,000, one can sequence their whole genome. But then one gets like ATGC, like a long strings of bases, and in fact, the bottleneck is in the understanding of all those variants. And this is also one of the reasons why we feel the fish can be helpful in that, because it helps us dissect the functional significance of some of, not all, but some of those variants. And that's where the bottleneck is right now.
Yes. Are any of the current technologies and discoveries that you mentioned, are they currently being applied for some treatments, or is it just at the lab level?
So, okay, so it's going to be a two-part answer. The one thing that's hard about aging research, the translation of aging research, and this is why a company like Calico, started by Google, is really trying to go at that, is that in fact aging is not considered to be a disease. And because it's not considered to be a disease, one cannot do a clinical trial on aging per se. And one has to choose, you know, like then a specific disease, and then you never really like address the questions. But to answer your question about the compounds that are like getting to the clinic, some of them are already, like rapamycin, because they were already FDA approved before, based on the fact that they are being used at high doses to prevent graft rejection. So for sure, at high doses, they are actually, I wouldn't recommend them because they will prevent an immune suppression, which is good for graft rejection, but it's bad in everyday life. But at low dose, and this is what people now, because it's FDA approved, then the trial can be more easily. And I was recently at a meeting where people from Novartis reported very good success with at least the vaccine response in the elderly. The flu vaccine is really badly, there's a bad response in the elderly. This was improved by low doses of rapamycin. So rapamycin is the example of the drug that's in the clinic already. Go ahead.
Are there ethical considerations about massively extending the lifespan? And in particular, how are we going to feed and house all these people? It sounds like a topic for a science fiction novel. If we doubled the lifespan, would there be room?
Yeah, so that's a complicated topic. And I was recently actually in Davos in Switzerland, where this topic was discussed, like compared to the environmental issues. And for sure, one can, people are trying to model actually the impact that like those compounds are like, you know, if everything works, would have. And in fact, it's quite interesting because all those, the research is kind of showing that basically when you, the research on longevity is showing that you increase the healthy portion of life. So in fact, they've done models, for example, the British government, and it concerns especially, it's true, like developed countries, but it's even true for countries that are developing. If you model actually the contribution to society of people because they are healthier longer, and then like the idea would be to square the curve. Like you make everyone healthier longer so that they can be autonomous, contribute to society, and have an amazing positive impact that because of experience that so far we already have, but we may have more of that, and then you drop dead. So that would kind of like be the idea, that the ideal situation. But what the horror scenario is indeed if you extend the decline portion of life, and then that becomes a problem. So, people are grappling with those ideas, and it is possible that some compounds will do one, some compounds will do the others, and it's very important to think about those different aspects or metrics and focus on the one that increases health span and active life and squaring of the curve compared to the ones that do that. Yeah.
They connected the young mouse to the older mouse, and I was wondering maybe the younger mouse, was it being prematurely aged? Actually, supposedly there's a bunch of junk in the bloodstream. I was just wondering, is there genetic junk floating around the bloodstream? Is there a way to look for markers that's happening in the blood that's collected,
Yeah, so those are good questions. So the first part of your question is, does the young mice hooked up to the old mice age? And actually it is. So basically what happens is that there is probably aging factors in the old blood as well as rejuvenating factor in the young blood. So when you hook them together, the young mice suffers from that. And that's a concern, when you think about doing a not even a blood transplant, just a blood transfusion. Who are you getting the blood from? I mean, of course, it's only once, but if people are doing repeated blood transfusion, that may be something to think about of which is the age of the donor for the blood transfusion. Now, as to answering your second question, Steve Quake here at Stanford has developed an exquisite assay for basically the genetic material that's floating around in the blood. So there is little, but there is some. So here in those experiments, researchers have excluded the possibility that it were cells, because there is also blood cells, obviously. So they've excluded that possibility because they did experiments to just inject the supernatant of the blood, not the cells themselves. And this still rejuvenates or age, if it depends which way you see you're doing the experiment. But that doesn't exclude free-floating genetic materials. And this is very recent that we have actually in our blood free-floating DNA. So it'd be interesting, actually. And I think this is one of the research area that Tony Whisker here at Stanford is doing. Yeah.
Hi, wonderful talk. I was wondering if the effect of exercise on longevity is understood, and if so, is it possible to convert that understanding into a treatment?
Yes, so people are very interested right now in exercise on aging because it's different than diet, for sure, in terms of mechanism. So dietary restriction will extend both maximal lifespan. It will make animals live longer in years, and then it will also extend their health and delay disease. Exercise does not extend lifespan, but it does an amazing squaring of the curve, and it has a rejuvenating potential that diet restriction does not have. So if you take an already old organism and you exercise it, cells in this animal, older animals, will rejuvenate, but it does not extend the lifespan. So people are trying to sort out the pathways that are activated by exercise compared to diet. Some of them are overlapping. The Insulin FOXO pathway is also, of course, regulated by exercise, but there might be more exercise-specific pathways. But that's new areas of research to dissect the molecular mechanism. So maybe in the next 5 years or so. Yeah.
I've heard that many, like newborns, are getting their stem cells frozen and saved. Do you consider that a positive preventive strategy?
I don't think it hurts for sure to save cells. First of all, it creates almost like a reservoir of cells, and that could be something. I mean, I think like, yeah, I don't see a drawback at saving the cells. It's hard to save them in a way that's perhaps can be used like, let's say, 80 years later, but that's probably a good strategy. Yeah, certainly.
What were the results of Siamesing the two young mice? Did they improve or did they stay the same?
So the two young mice and the two old mice paired together are used as controls, but because it is a little bit stressful, of course, to do the Siamese experiments on the too young and especially on the too old, actually. But those are important controls for the experiment to make sure that all the parameters are similar. People have studied the young pairs and the old pairs, and there is, after the little stress effect that they have of being paired together, there is no apparent change in them. So it's not the fusion of animals together that seems to be a factor. It's really the fusion of animals of different age and and factors that's in the blood of those different animals that seem to be acting. But I think it's an interesting consideration every time to sort of like understand also what the very procedure does. Absolutely.
So I have a question on hormones. You didn't talk about hormones at all, but I'm just wondering what you think about that role in aging.
You mean sexual hormone like estrogen?
Estrogen, progesterone, DHEA. I mean,
yeah.
Human growth hormone. I mean, just there's a huge decline, as you know, in hormone over
Yeah.
time. And I'm just wondering what you think about that.
Okay, so this is going to be a several-part answer because growth hormone. So actually, the gist of it is that all those hormones have transient positive effect, but very bad long-term effect on longevity. So they have a transient rejuvenating potential. So growth hormones makes people transiently feel better. Estrogen, testosterone also have this boost when people take them. But in terms of lifespan, animals of studies have been very clear that, for example, animals that have high growth hormone, either receptor or the hormone themselves, actually die much, much faster than other animals. And it is, in fact, the animals that have lower growth hormones that live much longer. But, you know, it's something to keep in mind. It's always this, there's no free lunch in a way. Like, it's always this amazing balance between, and exercise might be the same way, actually. One has to be trying to understand, you know, like when I said that exercise does not extend maximal lifespan, well, that might be the trade-off of exercise. Same idea, it makes one feel better and rejuvenate, but then in the end, at the expense of living a very, very long life. Dietary restriction is making someone live a very, very, very long life, but it is also at the expense of, you know, if you're on dietary restriction and you want to do something like really active and really, it's going to be tough. So it's always this tight balance between activity and longevity. And the thing is that people always thought it was like a all or none, but there's probably ways of you know, accentuating one, the other, having more cycles, in fact, rather than a permanent state. In terms of yeah, well, it is the big question that we're trying to understand. And in fact, it seems that it's like this, you know, like looking at the elephant and each person is looking at a different side, but in the end, it's all the same elephant. I mean, it's basically this story that aging is this very complex phenomenon, and people are looking at it from the stem cell side, from the molecular side, from this, and if each of them find something different, or not different, but they say, oh, I have the secret of aging or something like this. And in the end, and this is why now in science, people really feel that we need to go to the next level and adopt what's called a systems biology approach, because only when you have a systems biology approach can you put all those different pieces together and then really see the entire beast of aging as a whole and start understanding it. But we're not there yet. People are trying to do that, but we're not there yet, and certainly not for humans. Maybe closer to for worms.
Hi, just wondering, just out of curiosity, is your research currently working on like some kind of product to expand lifespans? That was weird. Or is it like too early to tell? And like, do you have some sort of idea for how that would work exactly?
Yes, so our research currently is more at the fundamental understanding of aging. But for example, the fish that we put together, that was an idea that we had that would allow us to screen more rapidly. And in fact, some of the pathways, because some of them we've been working for a long time on, so some of the discoveries that we've made, some of them are current, for example, for the Insulin FOXO pathways that we've worked a lot on. There are some drugs that affect this pathway, not created by us, but our understanding of the pathway has allowed basically, and others' understanding of the pathway has allowed some of the drugs to be tested. But we do not directly test the drugs. However, recently we've actually set up some collaboration where we are testing in our fish drugs that other people generate. So we'll see if they live longer.
What's your personal belief that, given the pace of advancement here, there'll be, in say, 10 to 15 years, benefits or improvements, discoveries that will make the average man in the street have an increased lifespan?
Yeah, so I think that if we look at 10 to 15 years, it's probably all the discoveries on the pathways that I talked about. Because things like rapamycin, metformin, resveratrol, those are all drugs, or LY. There's a lot of drugs that were actually already designed against this pathway. And the realization that those pathways are so important for aging can repurpose then existing drugs at just a different level or different dosage or different. So I feel like probably in the short term, that's probably by far, according to my personal belief, actually, at least what's going to happen. Because I feel like the stem cell aspects and those are probably further down the line. They have more risks as well. I don't know who has the microphone. It's probably like here.
Hi. Can you comment about the interaction of inflammation and insulin processing, sugar processing in the body, and how that affects the aging process and longevity?
Yeah, so that's a good question. Like, the inflammation has been recognized recently as one of the key hallmarks of aging, and in fact, it's actually predictive of death. So if one has increased level of interleukin-6, for example, it's a very bad sign. Like, that means kind of death in the year type of thing. Inflammation has a very predictive role. So inflammation does interact in cells, does interact with different pathways. So that's something important that people are. There's very little done yet on the interaction between inflammation and some of the pathway for aging, but that's an area of active investigation actually. So that's great. So probably that was the last question. So thank you so much.
Thank you so much, Dr. Brunet.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.